So the next presenter is a rare species. First of all, he's a nice Swede, and that is only something a Dane can joke about.
Yes, Finnish perhaps.
Maybe the Finns can do so. We can do that afterwards. The other thing is that Mats Velin, who is a senior director of biotechnology at the Swedish product agency, the MPA, attended the first meeting for biotechnology products back in 1995. And Mats is still a member of that working party. So here we have one who can explain what has happened in biotechnology for the past 30 years and maybe look a little bit into the future as well, Mats. The floor is yours.
Thank you. Oh, great. Yes, to start, I'm very honored to be invited to come here to try to give you some ideas of what has been discussed within the buildings you see on the right side over the last 30 years. So I start with this one, which is, title is Ad Hoc CPMP Biotechnology Working Party EMA, Westbury Circus, April 95. This was the first meeting of the BWP in London. I think someone has told me that that was also the very first meeting in a working party or committee at these new premises. And as far as I remember, I had the same tie. But that is more or less the only thing that hasn't changed. Or this hasn't changed. I don't wear this each and every day, but it's the same tie. But if we're looking at just this one, it's no longer an ad hoc group. It's no longer a CHMP group. It's a CPMP group. It's a CHMP. It's no longer biotechnology. It's biologicals, and it's no longer EMEA. It's EMA, and it's no longer Westbury Circus. So things has happened, but it's not only there, because when we see what we did discuss at that time, the BWP at that time had started in late 80s in Brussels, and there was a lot of discussion on viral safety of, in particular, plasma-derived products. So we had, in this very first meeting, there was long discussion on harmonization of blood donor screening, nuclear amplification techniques testing, virus validation. We had Creutzfeldt-Jakob's disease. There was some few hours of breakouts for discussions on dossiers. This is quite different today. Today is I would say 80% or so, maybe even more, is discussing on central scientific advice and new applications. And the time put to discuss the science is less at the meetings, but that is then handled by drafting groups where people who are really involved, which may also be more effective way of doing it, in the sense that the people that really know, and then it's told to everyone. So that is, so if we just go a little bit over what has happened then. So if we go even before 95, that you had the proteins, some of them came from body fluids or organs. You had insulin, you have human growth hormone, and then you were able to do it by recombinant technology. The first ones came in the early 80s, but we had the process defines the product, which had an impact on the requirements to do some changes to the production, because then, but you can't really show us that it's the same, so you may need to do clinical studies with your changes, which of course then will sort of lower the interest of doing the changes. But then we had you have an introduction of comparability for quality level through an analysis of physicochemical properties and biological activities, and showing that maybe you don't need clinical studies. We have an EU guideline in 2003. We have ICH Q5E in 2005. And then, then we had the first approval of the biosimilars coming into that. Yes, not only you do a change of your own product, you can compare to an originator showing that this is considered to be the same, and with limited clinical trials, you can get an approval. And the first ones we had was Omnitrop, was a human growth hormone. We had non-glycosylating, quite short proteins like less than 200 amino acids. Now we have much more complex proteins, and I'll show you a figure on the monoclonal antibodies that they are 1,400 amino acids. They are glycosylated and much, much more complex than we had. And what we have now is the discussion on the need for clinical data for biosimilars, depending on the and then I realized that this is not the last one of my presentation. Sorry, but I think it was no. So there were no need for clinical data without sort of tailored PK pharmacokinetic studies to be really correct here, and we do see some synthetic versions of peptides earlier produced by RDNA, and many of those that was discussed earlier this morning with 30, 40 amino acids are now possible to do by the synthetic routes. So if we have some milestones which we have gone through, that before EMA we had the classical CGD from human growth hormone. When during the sort of late 90s, you had new variant CGD, which was linked to the BSE outbreak due to changes in treatment of bovine material. That has a huge impact on medicinal products, that all products needed to be certified to be safe from a TSE perspective. And that, you could imagine that all of them, it was a huge work, and it was very well supported also by the European the Pharmacopoeia having their certification scheme. That meant that companies could come there, and then they had a certificate of their particular constituents, and that could be used for everyone that used that one. So it was really a useful one. Again, I have already mentioned biosimilars, which is quite a different change from where we were, and that is, I think it was triggered by Omnitrop, because that was initially applied as a well-established use, and then there was a discussion: is this a doable way? And in the end, it was a no, it's not. But then the biosimilar, or the similar biological substance, I think, or product, was introduced. And then one very important one is introduction of ICH Q8 to Q11. That is late 2000. That is a lot of instead of testing in quality, you have quality by design, you have criticality ranking, and other things that really go for what matters, and that has really changed the way of testing and looking at things, and say, okay, you may not need to test because you have shown that this is of no really importance, and having that together with, I would say, ICH as a whole has really been very important, that we are working and companies are working to different regions and coming up with a guideline that will be more or less acceptable by everyone is a great benefit and a good work. It takes a lot of work. Ask me or ask Jean-Louis, but it's and I think also ask Emer because you were involved earlier as well. But then we had 2009. We had the influenza pandemic. Everyone thought that this will be avian flu, H5N1, because that was what we have seen in China, and people were dying, but not that many. And so the guidelines were prepared. There was an administrative aspect, how to do it, that you submit sort of an core dossier or whatever. This is the way we are going to do it. But then when the actual strain comes, we are going to introduce that. So in that sense, we were well prepared. But then when it came, it wasn't an H5N1, which I think was a good thing in the sense that wasn't a killer that we would earlier expected it to be. So there was also, but in particular Sweden and others, there has been a handling of side effects post-pandemic on this one. Then we had a lot of very good meetings and workshops with industry and other agencies. And I think particularly like 2017, we have had a use of prior knowledge. And 2018, there was one on accelerated access that for unmet clinical need within sort of prime and breakthrough therapy in US. And the latter resulted in a joint guidance from us, which I think is really, really useful for companies in the sense that, yes, we can play the same game to both, and they are quite. And yeah, we have with the Brexit, as was mentioned by Guido, and the movement of London to Amsterdam, of course, had some impact on our work, concentrated more or less totally on running the applications and so on for very good reasons. And then 2 months, I think it was after coming to this building, you had the COVID. And you have really to change the work. You couldn't come here. You had to work in a completely different way. We saw some new vaccine modalities, and we could really discuss acceleration there because the efforts put down on approval of products is marvelous compared to what we have had before. And that is, then you could say, yes, these two workshops that use of prior knowledge and also that what could be acceptable in an accelerated situation where you don't have anything for that clinical need. So we were actually quite well prepared for that. So just going through some of the types. So the first monoclonal was approved in Europe in the late 80s. It was produced in mice ascites. It appeared quite, with the methods at that time, it appeared quite pure. It wasn't. The second one came in 94. There were two more in 98. 2005, we have 16 monoclonals. 2015, we have 52 and one biosimilar. And 2025, we have 210 monoclonals approved in total, of which 61 are biosimilars. And 12 of them are then also antibody drug conjugates, and where you link a chemical drug to a monoclonal to really have a very high specific therapy. And why is that? I think initially that, yes, you can do it and you can direct it somewhere, but you didn't really know how to make a therapeutic use of it. It was used to some extent for diagnostic purposes with some radio labeling, but then more understanding of what can they do, and also we had improved analytical methods, which gave some meaningful comparison between the biosimilar and the originator, and that is why we are seeing this huge number of new biosimilars coming up. For vaccines, which is close to me, I think this is by far the biological medicinal product that is given to most people. And it's different in many other products because this is given to healthy people. This is very often given to children and healthy children. And safety aspects is therefore very primary here. And if we would have difficulties there and the trust in vaccine would be poorer, then we would be out on thin ice, I think. So it's interesting, we have a very long history from the late 700s, but what we have now, we have combination vaccine where you can combine antigens from multiple infectious diseases like diphtheria, tetanus, pertussis, polio, hep, or you have antigens for different serotypes of the same bacteria. And we have, I think for the moment, we have 21-valent pneumococcal polysaccharide conjugate vaccines for the time being. We started with 7. We have virus-like particles for the papilloma vaccines. We have the mRNA vaccines, and for example, then 2 of the COVID vaccines were there. And then we also had the adenovirus modified to contain a gene to making a specific protein. So it happens a lot. We are working a lot with new adjuvants and so on. So this is a very interesting field, I would say, which is also the same thing for cell and gene therapy. That, yeah, I'm not going into the background, what they are, but what we see is that a lot of things now come, which is a combination where you put in the sequence you want into a cell, and then it can be express a new gene, so and then given back. So this is really a game changer. That all of a sudden it's possible to treat what in the past used to be untreatable diseases. And I had a look at what is then for Prime. Currently there's total 79 products. ATMPs is 35 of these. Biotech, other biologicals are 25, chemicals are 19. It's really that with this type of products, we can really medicate a number of diseases we couldn't some years ago. And we see a similar scenario for the approval as for the monoclonals. The first one was approved in 2009, next one in 2013, 2 more in 2015, and now we have another 19. So it happens a lot. And what is then to be expected in the future? I think a lot. We will see more of the ATMPs for good reasons. We would see personalized medicines, including 3D printing. We will see the antibody drug conjugates even more. We will have point of care, bedside manufacturing, where the final part of the preparations will be done close to the patient, and that will be in particular ATMPs. We are making use of models, digital twins in control and development of new products and their processes. And this may speed up and lower costs for development of new product and improvement on old one. And we also see, as we said, I think it's clear we will expect more mRNA products as we have until now. So having said that, ATMPs and antibody drug conjugates and some of the others are quite expensive drugs. And that is, so the case is, yes, there is something, but is it possible? Is it at all possible? Will this be used? Because the treatment per year is really high, but let's see what can be done. At least it's there. We'll see what will come. And then, of course, there's a lot more, but we don't know yet. It may be that Marco will have some idea in his presentation, but I think, or I know that this was the last. So thank you very much.
Thank you so much, Matt. Would you be okay answering some questions if there would be someone in the audience here? While audience think, I will ask you one. You raised the issue about affordability of ATMPs.
Yeah.
But you also mentioned biosimilars. Will we have biosimilars to ATMPs?
That's a very good question. I guess the biosimilar, in the sense that biosimilars themselves had evolved, that earlier on you could do 150, 170 amino acids shown this, and non-glycosylation. Now we do it for there. We need to have a better understanding what exactly in these ATMPs are the important one and how can we do it. I think we have a number of them which are quite comparable, but they stand on their own legs for the time being. So maybe in the future, but it is a tricky question, because it's also that companies are putting a lot of money in it, and some they are not coming to it because they are stopped in phase 2 or 3 or so. So it's a lot of money put into it, and how can we do it? So it is something we need to consider, because I know that we have said, okay, this would be perfect for this patient, but the regions cannot afford it. So that's a really issue.
Any question in the room for Mats? Otherwise, we will thank you so much, Mats.
Thank you.
Thank you.